Okay, we're gonna get going. So thank you all for being here. We've got 40 people on the line. That's fantastic turnout. Friday morning meeting like this. But no, this is fantastic. I don't know if we're actually gonna take the full time today, but we really kind of want you guys, everyone to kind of think about what we're talking about today, and we were encouraging lots and lots of questions because what we're trying to do, we haven't really fundamentally done here in Alberta. So I'll do some quick introductions. So I'm Megan, and I am with the Alberta Invasive Species Council. I've got Paige with me here. She's with the AISC as well. Paige, of course, runs our biocontrol release program for the agents that we oversee here, and she's going to talk a little bit about that in a moment. Uh, we have Erin Foster, who is joining us from Wyoming, who is part of a consortium for biocontrol in that state. And so what we're trying to do is kind of similar to what they have going down there. So Erin's just going to give us a rundown about how that works in their jurisdiction. And then we've got Phil all the way from Switzerland. It's 5 p.m. He's very kind to come do this for us at 5 p.m. his local time. And Phil is a colleague of ours that we work with at CABI, which is, I can never remember, but it's the Center for Agriculture and Bioscience International. And we've been working with CABI for a while and contributing a small amount of money to help support the Comintanzi biocontrol research. And they do great work over there. CABI is kind of where the biocontrol agents come from. So if we kind of want to be a part of seeing and prioritizing new agents, we really should be working with Phil and his team over at CABI. So the goal today is really to provide an overview of what is biocontrol. Phil's going to talk to us about the ins and outs and what they do over at CABI. And then Paige is going to talk a little bit about the current status of biocontrol agents and availability in Alberta. Erin's going to jump in and talk a little bit about how the consortium in Wyoming works. And then I'll give you a little bit of a breakdown of what's been done so far in Alberta towards the development of a biocontrol consortium and kind of talk about some next steps. So we are recording the meeting this morning, and we're going to be posting that on our website for future reference. And we will try to put together a workspace for anyone that wants to be involved in this consortium. So we'll be pulling all of the information together, and it'll be made available to you as well. So with that, I think that we can jump in. I think that we can jump right in, and I'll stop sharing my screen. And we definitely welcome questions. So while folks are presenting, folks, you can type into the chat, or at the end of each presentation, there'll be an opportunity for questions as well. But I'll pass the floor over to Phil. So the floor is yours, Phil.
Thanks so much, Megan. Is the sharing looking good?
Looks great.
Okay, perfect. Yeah, thanks very much. Sorry, I can't see, for whatever reason, I don't see any kind of images. So just jump in if you need me to slow down, or if you have any questions. Yeah, feel free to interrupt me at any time. So thanks very much for the opportunity to speak to you guys today. I'm going to talk to you a little bit about the ins and outs of classical biocontrol. If that's a bit of a recap, for some of you folks, that's not a bad thing. And if it's new for other folks, that's even better. Okay, so for those of you who are not aware of who or what CABI is, so we're a not-for-profit intergovernmental organization established at a United Nations treaty level, through an agreement. We're not owned by a single entity or country. Our member countries, of which there are 49, have an equal role in the overall organization's governance, policies, strategic direction. And we deal with issues of global concern, such as invasive species, which is the theme that my team is under, and then food security and food safety, and that's largely through our research and international development. We have a second branch away from the research, which is essentially publishing. So we publish scientific information in the form of books, ebooks, the invasive species compendia. If you haven't seen those, I urge you to go and check those out online. These are our member countries currently, and what's kind of cool with this list is that there's a whole range from kind of developed first world countries such as Switzerland or yourselves in Canada, all the way through to developing countries. So we get a whole range of perspectives in terms of food security and invasive species research that we can do. Okay, so just to give you a very brief kind of recap on the partnership between CABI and Canada. It's been going on for a long time. And actually, our center in Switzerland was started in 1948 as a substation of the Commonwealth Bureau of Biological Control. So we've had just last year, they celebrated the 75-year CABI Canada partnership at the ESA meeting. And in the early days, the center was largely set up to study forest pests, particularly those native in Europe. But with time and shifting priorities, that's now changed to primarily agricultural pests and alien invasive weeds, almost exclusively. So this is a really cool kind of aerial image of CABI. So you've got the building in the top left of the picture, and then we've got various garden beds to do our experiments. We've got some cages. We get aliens visiting with crop circles, so we can do some pretty cool open field tests. And we are based in Switzerland in the kind of northwestern corner. So we're very well suited for a lot of the invasive plant species that are invading Western North America in the United States and also in Canada. A lot of your invasive weeds come from Europe and Eurasia. Brief introduction of the team led by myself. So we have four scientists, four permanent scientists sitting in the CABI Center, Sonia, Patrick, Ghislaine, and Laureline. And then Ivo is a scientist that sits in Serbia. Cornelia keeps our lab running. She's our brilliant lab technician. Florence and Julie are our garden technicians. And then Harriet is our kind of honorary member that we lean on for advice. She has a huge amount of experience that we like to use. And then every year we get between 7 to 10 temporary research assistants, kind of summer students or summer interns. And often those are there'll be between 2 and 4 Canadian students. So we keep that connection going there as well. So a lot of our work at CABI Switzerland revolves around developing biocontrol. So we do foreign exploration for potential natural enemies within the native range of the weeds. We assess risks, so like host range testing, essentially trying to determine whether those species are safe. And while doing that, we're continuously trying to improve protocols for testing to ensure that we stay ahead of the game in terms of efficiency and investing in kind of biocontrol winners, if I can call it that. In addition to that, we also look at impact assessments of potential agents, either on the target weed that we're interested in, or potentially on non-targets that we worried may have sort of off-target feeding. Yeah. And ultimately, our goal is really to get those agents released. So we play a key role in developing the petitions for field release with our partners, certainly in Canada. That's the way it works. And so we submit that document and hopefully get the agents out on the ground for everyone to use. So this year in 2025, we're currently running 21 projects, of which 20 are for North America. We have one co-financed for Australia and one for South Africa. And kind of what we're here to talk about today is most of these projects are financed by sort of consortium approach. And what that means in many ways is we have multiple donors per project or per weed species. And this really ensures continuity and longevity of the projects. Some of these projects, as I'm sure Aaron will point out, we've been working on for the last 25 years. And we're just getting to the stage now where we are in a position to start petitioning for agents. So some of these projects do take very long, but typically we're looking at between sort of 10 to 15 years of testing. And what this also means in many ways is it's excellent value for money for individual donors. So, for example, if you were contributing $5,000 or $10,000 per year to a project, you might be getting a product that's sort of worth in the $50,000 to $60,000 range once we put all the donors together. So this is the long list of weeds that we're currently working on, and I don't expect you to remember these at all. But if you are interested in finding out more information, my email's on the bottom. feel free to reach out. Megan will have my email as well. I'm happy for her to send that out to everybody in case you miss it here. I do just want to point out the three weeds at the bottom that I've circled there. These are kind of our new kids on the block. All three of them occur in Canada. Echium vulgare or blue weed is financed from BC at the moment. And then we've got Hydrocharis or European frogbit and water soldier, which are both in the eastern part of Canada. But that's so far they're only funded from the Army Corps of Engineers in the US. Okay, so biocontrol of weeds, what exactly are we talking about? So when we talk about classical biological control, we're really looking at the deliberate use of specific natural enemies, and specific there is quite an important word, to control an exotic weed. So we typically wouldn't be looking at kind of biocontrol options or kind of classical biocontrol options for native weeds, but typically focused on invasive alien or exotic weeds. And if I can just put this in a kind of animation, if you will. So what you often have is, if this is the same plant species in the native range, typically they're quite small. They might be quite scraggly, usually not in monocultures, but quite sparse on the landscape, not necessarily causing a problem. You have the same species in the introduced range. Often they're bigger. They have more resources to put into growth and not so much into defenses. And they form these monocultures, and essentially they're problematic. We don't want them in our landscape. Now, in both ranges, what you'll often find is a suite of generalist herbivores feeding on them. These might be grasshoppers or kind of lepidopterans. Usually, they may cause some damage, but they're not really population level regulators. The difference in the native range from the introduced range is you also, in addition to the generalists, you have this kind of suite of specialist natural enemies. And what we try and do with biocontrol is to reunite the specialist natural enemies with the invasive plants to kind of reestablish that natural ecosystem. And we try and do that obviously in a safe and effective manner. So if you've been to any biocontrol talk, I'm pretty convinced that you've seen this diagram. And essentially what it is just putting what I just said in kind of graphical format. So you've got a population density of a weed up here, and there would be some sort of damage threshold, and this can be either an economic threshold. So the weed might be impacting, say, cattle farming, or ranching, and that's having a direct economic impact for farmers. Or it might be a kind of ecological damage threshold, which, it's maybe affecting native pollinators, and that's then having knock-on effects on the native ecosystem. So typically the weed density is above that damage threshold, which is why we're all here talking today, because we want to bring that density below that damage threshold. So it's not causing economic or ecological damage. And of course, we want to try and at the same time restore native species communities. So what we'll have is, once we've developed a biocontrol agent, we would release that agent into the system. That agent population would increase because there's no resource limitation. The weed is everywhere and highly abundant. And what we'd like to see is that over time that population goes down. And then, of course, there'll be either annual fluctuations or seasonal natural fluctuations in the population, which is why we have this kind of predator-prey cycle going on. And then obviously what we'd like to see in the system is this desirable vegetation going up. So just to give you a couple of Canadian specific examples where biocontrol has been incredibly successful. I really love before and after photos. I'm always a little bit worried that it creates a kind of unrealistic expectation, but this biocontrol can really achieve this given time. And if you contact Paige, she'll get you agents, and you will start seeing this on the landscape for several of the weed species that you work with. So St. John's wort was a huge problem in both Canada and the US, and completely covering the landscape. Chrysalina is the biocontrol agent that was released in the 1950s. And if you have a look at this picture over here, so 1949, you got this chap standing there. The landscape is completely covered in St. John's wort. Roughly 10 to 15 years later, you're looking at the same the same area, and St. John's Wort is essentially rendered insignificant on the landscape. There was some data collected with that, and what you can see here is we've got a weed density and the insect density increasing, so once released, and then increasing in the landscape. And what you can see is that weed density completely crashing, the ideal scenario that we're looking for in weed biocontrol. So there's more than one example in Canada. There's loads of examples, but these two are just sort of two very impressive examples. So this is Mogulones crucifer, which was released against houndstang, and I'm sure you know what houndstang is. It's a weed of forested rangelands, and this is a photograph prior to release of the weevil. And chemical control is quite limited. It has massive impacts on the landscape. It outcompetes native vegetation. It pulls native pollinators away from native plants. It's poisonous to livestock. It has these awful burrs that kind of stick on. If you took your dog for a walk through houndstang, I'm not sure you'd have much dog left. It'd sort of just all be covered in these sticky seeds. And if we look at 2-year cycles, so Mogulones crucifer was released in 1997. 2 years later, so that's pre-release. 2 years later, you can already see kind of ground opening up and the population of the weed going down. And 4 years after release, houndstang is basically completely removed, or it's rendered insignificant at that site and at a landscape level. So absolutely fantastic biocontrol agent. But how do we get there? So how do we get from having a weed on the landscape to developing a biocontrol agent and then actually getting it out on the ground? So there's essentially some basic steps that we follow in weed biocontrol, and it all starts with identification of the weed, and that's both in the invaded range. So what are we actually dealing with? Say, for example, in Alberta, are we dealing with one weed species? Are we dealing with multiple species? Are we dealing with particular kind of genotypes that we might be interested in? So we spend a lot of time trying to figure out what we're dealing with in the invaded range, and then what we're dealing with in the native range as well. Once we kind of know what we're dealing with, we look at the life history, and that can be, from biology through to origin, distribution. And that really allows us to figure out where that sort of Achilles heel of a particular weed is. So, if you're dealing with something like Russian olive, you would be thinking in a very different set of terms compared to houndstang, for example, which is a biennial plant which only lives for 2 years. You might be looking for very different types of agents and very different modes of action. Then we survey for natural enemies, and that essentially starts behind a computer screen. So looking at the literature, looking at iNaturalist, looking at herbarium and insect collections, and then actually getting boots on the ground and surveying the native range, looking at what's actually kind of exerting that top-down pressure, and whether we can find something that may be considered specific enough to take forward. That takes me to the selecting of that potential agent. So we want something that's, looking at the weed's life history, something that's going to be damaging enough as well as specific enough. And then we look at the agent biology and efficacy, followed by the risk assessments and the host specificity testing. And then once we've deemed that agent specific enough, as I mentioned earlier, a petition for release would be developed where we summarize all of the data, proving that this potential agent would be safe for release in Canada or in North America. That then gets submitted to the regulatory bodies, which starts out with the Biocontrol Review Committee, which is a group of experts that review the data, make a recommendation, which is then submitted to the Canadian Food Inspection Agency, or the CFIA, who then make a final judgment and decision whether to issue a permit for release or not. Fortunately, that process in Canada is super smooth, a really well-oiled machine, and we can typically get agents from when we submit a petition to a release permit within a year, if not sooner. But some have taken up to 2 years, which is quite different when we think of the American situation, where there's many more steps or many more hoops to go through. and that can take anywhere between sort of 2 to, gosh, I mean, even up to 5 years. We're still waiting to get through some of those hoops. Then once a permit for release has been issued, we then look at mass rearing, release and post-release monitoring. Cabby, a lot of our work is very heavily involved in the first part of that, and then we would hand over to typically to partners in Canada who have quarantine facilities. So those would be AAFC partners. Rose de Klerk Float is or now that she's retired, she's still involved in our work, but not officially. Would then be, the kind of the first ports of entry, so the quarantine facility, and then they would then rear and make releases and do the post-release monitoring. And then Paige will be talking to you about the agents that are sort of currently available in Alberta or that they deal with in Alberta. Okay, so when we talk about host specificity, we're really talking about specialization of insects, and this really happens. It's not a simple process. It's really a intensive plant-insect interactions that happen over time. And when I talk about over time, I'm not talking about sort of 1 or 2 years or even our lifetimes. I'm talking over thousands of generations. And it involves more than a single step. So it involves several sensory, physiological, and behavioral processes that are incredibly robust. So they're very difficult for us to pin down often. But what that means is that they're also very, very robust and unlikely to break down very easily through sort of evolutionary processes. So they're relatively stable within a population. And if we think about host specificity within insects, we can break them down into 3 broad categories, or bins, if you want to call it that. So we've got polyphagous insects, which feed on just about anything green, which interestingly, there's very few insects that actually fall under the polyphagous group, but there are some that are quite damaging. And those are obviously not good biocontrol agents. We're not really interested in those at all. What we are more interested in is the oligophagous and monophagous species. So oligophagous would be those species that feed on kind of a relatively small subset of species within one genus or potentially within a couple of genera, but typically not spreading outside of the tribe of plants that we're interested in, or monophagous species, which are really targeting a single species or single target species. So just to run through a couple of definitions, in terms of host specificity testing, we talk about the physiological host range very often, and those are the plant species on which a herbivore can complete its life cycle. And that's very different when we think about the realized or ecological host range, which is what we actually see in nature. And that the definition of that is really plant species which are utilized under field conditions. So although a plant may be within the physiological host range, it may actually never be at risk once the agent is released. And in many ways, what we do is with our host specificity testing is we try and define both of those. And by starting with the no choice testing, that really gives us an indication of what plant species are within that physiological host range. And that's usually very conservative and can give us an indication of where risk may lie. So essentially what we do is we expose the insects to a single plant species. The insect either feeds or starves to death, or lays an egg or just really cannot. If those plant species are then are within that physiological host range, we then move to kind of trying to define that realized or ecological host range through dual choice tests where we have 2 choices between the target species and a non-target species, multiple choice, which is a lab test, but trying to approach a kind of natural or kind of natural ecosystem. And then if those plants are still utilized under those conditions, we then really try and go to the open field and really work outdoors where the insect is not caged, it's not held back in any way. They're free to fly in, they're free to fly out, and to either utilize or not those plant species. And that's really the gold standard of host range testing, and allows us to really pin down that ecological host range. So just to again kind of put that into context. So if you imagine that big green bubble being the physiological host range, the realized host range is always a subset of that. The realized host range can never include a plant species outside of that physiological host range. And the possible reasons for this shift might be steps in host selection behavior, which act as ecological filters. If an insect can't smell a plant through the volatiles, there's no way it would ever find that in nature. Or if it doesn't perceive its kind of color profile or anything like that. We might have more acceptable hosts always available, or the insect is very good at dispersing, so it always finds more acceptable hosts. Or there might be a complete lack of coincidence. If we're talking about a seed, a flower, or seed feeder, if your plant in question, say, flowers in autumn, but our insect is active in spring, we kind of have that that temporal separation. Or you might have a spatial separation where there's no overlap in distributions. So classical biocontrol has an incredibly positive track record for identifying safe agents. We've, over the years, so for the past 115 years that we've been doing weed biocontrol, we've done an incredibly good job at identifying safe agents. So we've released roughly 500 agents globally since the initiation of weed biocontrol over 100 years ago. And we've had a 99% success rate in predicting non-target host use. In all management options, there's always an associated risk, and what we need to do is to try and weigh those risks and benefits. So if we can identify which potential non-targets are at risk, it allows regulators to then determine whether that risk is worth taking, and that goes with any control method, not only classical biocontrol. When you think about herbicide applications or manual removal or mowing, there's always going to be non-target risk associated with that. But it's at what level of risk are you willing to take? Of all of the agents released worldwide, only 3 cases of all intentional releases actually threaten populations of non-targets. So that's less than 1% of all biocontrol programs. And all 3 cases, this was predicted prior to release. But the benefits of releasing the agent outweighed the potential risk to the non-targets. So just to reiterate, classical biocontrol has an overwhelmingly positive record for safeguarding native and important plant species. Okay, so just to give you guys a bit of a sneak peek on what to hopefully expect in the near future. So some petitions that we have on the horizon. So we've got agents for common tansy, which will be or have been petitioned, for Dyer's woad. For flowering rush, we have a new agent which will be petitioned quite soon, this leaf mining fly. And then we've got for oxeye daisy, for hawkweeds, Russian knapweed, and for toad flaxes. So a lot of exciting stuff coming up on the horizon. In addition to the agents that Paige is going to be speaking to you about shortly, there's also some kind of new agents that are kind of hot off the press, if I can call it that. So we've got Bargus nodulosus, which was recently approved for release. We've got the seed-feeding weevil for garlic mustard, Soterincus constrictus, which has recently been approved for release, and then a whole suite of other agents. Again, if you want more information, I'm happy to provide that. If you just drop me an email or reach out to Paige, I'm sure she's aware as well. And that's what I have for you. Thank you very much, and happily take any questions if you have. And then my email's in the bottom left as well.
Great. Thanks so much. That was fantastic. There's folks still joining. I have some questions. My first question, and this is one I've had for a long time. You can see us now.
Yeah.
So my first question is, why weevils? Why are weevils always like, there's so many weevils that are they generally specialists in nature? Is that the idea, or are there other factors?
So weevils are a massive group within the Coleoptera. So there's hundreds and thousands of species of weevils. They're typically very damaging, as you'll know, because there's a lot of crop pests which are weevils as well as biocontrol agents. And there's a lot of specializing within the group. So even if you consider some of the crop pests like the Citrinchus obstrictus, which attacks canola or rapeseed, that's although it's a pest on a crop, it's also a specialist within the Brassicaceae. So you're not going to find it on a bunch of other species. And they're incredibly they're just super cute and cool to work with and really damaging and typically very specific. There's possibilities of finding monophagous species. We have species that may even attack only certain kind of lineages or populations of a weed. So they're a fantastic group to work with.
Yeah.
I get very excited about weevils and yeah.
Love it. Love it. So you also, so folks, if you jump in your questions and feel free to unmute yourselves or you can type into the chat, whatever you'd prefer. But I have another, and I'll just keep asking my questions until. So you talk about classical biocontrol. What is non-classical biocontrol? Is that like gene editing and stuff, or like what?
So I mean, there are, yes. So this is actually under debate at the moment, what the definition of biocontrol actually is. And
Okay.
so when we talk about classical biocontrol, we're really talking about reuniting the specialist natural enemies with an invasive population. To try and bring back that kind of there's a bunch of different hypotheses why invasive species become invasive, and one of them is the kind of enemy release hypothesis. So what we're trying to do is reunite the specialist natural enemies to bring back that top-down pressure. So when you talk about classical biocontrol, it's really referring to that. There've been kind of iterations. I'm kind of a bit traditional, so I don't really like changing terminology because we're so like if you talk about classical biocontrol, the biocontrol community understands what that means. But there's been talk about changing that potentially to importation biocontrol, which might be more descriptive.
Okay.
Whether that gains traction or not, we'll wait and see. The jury's still out, in my opinion. But then you've got other types of biocontrol, like, for example, gene editing, or you have augmentative biocontrol, which is trying to increase populations of a natural enemy already existing in your ecosystem. And that can be that could even be a classical biocontrol agent, or it could be a native herbivore that you try and increase its population. There can be cultural biocontrol, which is kind of looking at managing your ecosystem. So improving pollinator health has knock-on effects for the whole ecosystem, for example. And that will help kind of restore that resilience within the ecosystem. And then there's gene editing, which I mean I believe what the guys are doing is safe. I just don't understand it, and yeah.
Me neither. Okay. And I have another like really random specific question, but who's funding the blueweed biocontrol? Can you do you, what? Yeah.
BC, British Columbia. Yeah.
Okay.
So BC has been Canada's or the Canadian sort of biocontrol powerhouse in terms of funding and getting new projects off the ground. We're getting an incredible amount of support from them, and quite often, blueweed is one of those weird ones where like everybody's like, "Oh my God, you guys really should work on blueweed," and you're like, "Yeah, we really should," and then it's like, okay, so to start, we need money to start working on a project, and like once you have a little bit of money, like then everyone's like, "Oh, cool, actually, we could," and it kind of it gets that snowball effect and gets things moving.
Yeah, no, absolutely. I can totally see that for sure. And some of the folks here on the line will know that I live in a municipality in Alberta, and I always joke that blueweed should be our municipal flower because we're like the blueweed capital of Alberta. So I feel like I need to rally locally here to get some funding to help support that. Anyway, Rick will probably be happy about that.
And we've got two agents in culture which are pretty cool. So we've got a weevil, which is very nice, a root feeding weevil,
Okay.
Mogulonis geographicus.
Okay.
It's got a very cool pattern on its back. And then an aerified mite, which has turned out actually to be a new species. We're still trying to figure out what we see a very cool damage, which is just like a total kind of witch's broom on the flower. So it just 0% seed production, like nothing.
Wow.
I'm not 100% convinced that's actually the mite doing that. But Sonia, the scientist working on it, is sure that every plant she's found has the mite on it. But in my opinion, the numbers are not high enough. So the jury, yeah, we're still we're proving that this year. We've both been keeping plants at home and with mites and without mites, and we're going to see if we can figure this out.
Well, keep us posted.
Yeah,
Yeah.
absolutely,
Okay.
absolutely.
Awesome. Okay. Does anyone else have any questions before we move along? Okay. Oh, here we go. We have a question. Uh, have there been any concerns associated with leafy spurge flea beetles, species control of leafy spurge host specificity? So Trey, I think that's asking about whether
Off
they've gone off
target.
target, I believe, but please clarify, Trey, if I got that wrong. So, Bill.
Trey, not as far as I'm aware. It's been a bit, so the leafy spurge biocontrol has kind of been through a few ups and downs. So it's an incredibly old project. I think some of the work was done in the 1950s and 60s. And taxonomy has changed a lot. And what they were actually collecting potential agents from, and so essentially with genetics we've been able to really nail down what the species are now in North America, and some of the agents that were collected were not actually from those species, but I don't believe that there is any non-target attack or non-target host use by any of the agents in North America. I think leafy spurge is sort of a little bit on its own in terms of taxonomy in North America, and kind of having agents that are not kind of more in that oligophagous bucket and not in the monophagous bucket was actually quite helpful for the biocontrol program, and didn't actually result in any off-target host use.
Thanks, Bill. Okay. Any other questions? And then we'll have time for questions at the very end too. So folks, don't be shy. It's a safe space. You can ask any questions. Okay. All right. So I think next up I've got to, oh, and Paige, you have the presentation on your end. You'll share your slides. Okay. Okay. So we're going to switch things up. We're going to pass it over to Paige, and Paige is going to talk to us a little about biocontrol in Alberta and the current status and availability of those agents. And yeah, over to you, Paige. Thanks.
Thank you. Is this in presenter mode? I hope so.
It's perfect.
Okay. Thanks, Phil. You've covered majority of what we need to talk about. We have all the biocontrol done, so we can dive into the release program that we have with the AISC. So we started in 2001 as a collaboration between the Weed Biocontrol Research Team at Agriculture and Agri-Food Canada. They do kind of the research and the testing, and then it was also a partnership with several agricultural fieldmen that did all the releases and the field work. So it was a really unique program, and it was entirely funded by participants, and now it's operated by us at the Invasive Species Council. So as a service for invasive plant managers to purchase these agents in conjunction with our very expert consulting. The first target species for this program was leafy spurge. And since starting with leafy spurge, the program has expanded to working with over 39 different organizations. I'm sure that's higher now. So of course, whether that be you guys with the municipalities, individual landowners, all the way to Alberta parks. We got a call from Manitoba recently, but we supply biocontrol agents for their invasive plant populations. So from this kind of ongoing support and over the years, we've been able to offer additional agents for release. So that includes houndstone. We don't offer it anymore. We also have Dalmatian toadflax and spotted diffuse and Russian knapweed agents. So since starting this program in 2001, we've released and monitored agents at over 1,500 locations in Alberta, and that number definitely continues to grow. I think we have 70 or so on the docket for even just this summer. This is a close-up of what the agents look like that we offer. We have spotted and diffuse knapweed root weevil on the top left here. like Phil mentioned, they're pretty cute. I never thought I'd say that about a bug, but this guy is. He's definitely our largest one as well. And then we have the knapweed seedhead weevils on the top right. So those seedhead weevils will lay eggs in kind of the flower heads, and then the larvae eat the seeds. The adults kind of feed on the leaves. Then with the root weevil, those guys lay their eggs on the root crown, so just below that soil surface, and the larvae will actually tunnel through the taproot and expose it to infection. So this is cool. These agents can be used together, and as they attack different parts of the knapweed and will not actually compete with one another. I'm not going to butcher the Latin names for any of these species, but I can put them in the chat if you're interested. We also have Russian knapweed gall wasps. They lay eggs in the plant stem and they create a tumor-like growth or a gall, which draws nutrients away from the plants and reduces their ability to grow. We have, of course, leafy spurge beetles. We release two types. There's a brown and a black beetle. The black ones are on the screen. These beetles feed on the leaves and flowers of leafy spurge, limiting the plant's ability to grow. They kind of create like a skeleton effect on the leaves or bruising on the stems. So these will hatch and burrow in the soil where the larvae overwinter and feed on the spurge roots, which is definitely where we want the majority of the damage to come in, and it does, until they emerge in the spring. Then below we have the Dalmatian toadflax stem weevil, of course, another weevil. They leave a distinct shot hole pattern on the leaves and stems after emerging that stunt the plant growth. So in that picture there, you can see all the holes and the tops kind of droop and yellow. That is all the agents that we have. To collect these guys, we use nurse sites or the original release sites to collect the biocontrol agents. So none of our agents are actually, that we release are actually reared in the lab. All insects belong to the Crown, not the landowner, but we still, of course, need approval for using their land and whatnot. So there's lots of logistics associated with these sites. Of course, the landowner cooperation, kind of the location in the province, the amount of the invasive plant itself, lots more. Essentially, each agent comes up at different times in the growing season. So early spring, late summer, and we wait until there's a boom in the population to collect from a site. So for example, with leafy spurge beetles, they'll be up early to mid-June. So we don't really collect orders until late June to mid-July. Depending on the site, of course, because east to west, the plants will come up earlier. So if we have an east site, we'll collect east first and then move our way west. We don't take all the insects from a site, and so we allow there's opportunity for the population to continue every year. Every few years, we'll try and move to a new collection site so the previous one can kind of recoup their population and really get to work on the area for plant control. The Invasive Species Council charges for orders of these agents, which is essentially just recovers the cost of travel and time in the field for us. Yeah, and costs haven't changed for about 5 years since I've been at the program, potentially longer even. But $900 plus GST, and that's regardless of agent that you select. So depending on the agent, we collect them using either a sweep net, we clip the galls, or we actually hand pick them off the plants themselves. And we'll collect kind of a specific amount to release at a site and that's deemed appropriate to start a new population. So if we said Russian knapweed gall wasp was your order, we'd have 400 galls that we've clipped, whereas an order of leafy spurge beetles is about 2,000 beetles. We don't individually count those. There's a measurement on the jar, and we try to hit that line. Release sites depend again on the invasive plant. The invasive plant, but overall, most releases will be an open site, kind of away from tree canopy, shelter from the winds, full sun, a patch of about 20 meters squared. We try to avoid floodplains as some of the insects overwinter underground, and usually away from herbicides. Some of these agents, again, are very sensitive to them. Many of the agents will become self-sustaining and provide ongoing control, though some may require repeated releases as well. Orders for this season have now closed. I put the date at the end of April, but if you're interested in leafy spurge or any of the spotted and diffused knapweed weevils, definitely shoot me an email because the collection period for those guys occurs later in the season, so we can definitely squeeze those in. I put a picture of one of our informational sheets on the right side of the screen. It's just a quick few pages kind of identifying the target species, biology of the biocontrol agent, and really a quick guide on how to prepare for a release and how to move the agent once they've established. So we have these how-to sheets for each agent that we release. We usually give them out to landowners that aren't that familiar with biocontrol. We do have some other biocontrol agents coming down the pipeline or older ones. Houndstone, as you mentioned, was very successful. We stopped releasing this one in 2021 as the populations of the plant already had this insect present, which is crazy. So they're self-distributing, which is amazing. We have scentless chamomile and fungus for, Canada thistle. And Chris Saunders from Strategic Weed Management does the releases for those guys. So we can provide his email as well in the chat later. Yellow toadflax gall mining weevils were reared in the lab, but this stopped a few years ago, and initial releases have shown actually quite significant reductions in the toadflax sites that were released in 2015, although they did notice less than 5% wasp parasitism. But nothing that impacted the agents too much. So hopefully we can start distributing those guys in the coming years. I think they're still looking at them. Early releases of Russian olive gall mites and oxeye daisy stem moths in BC and southern Alberta from 2023 actually survived our -40 extreme winter conditions. So that's great news. And then rearing continues for further releases and monitoring of those guys. So again, hopefully soon we'll see those ones. Then common tansy in development right now. They're ready for releases this year pending CFIA approval, but they're not yet ready for distribution to counties, but really just to establish those nurse sites or populations. So that is basically my super quick update that we got from Agriculture and Agri-Food Canada about the upcoming agents, and really majority of what I have for you guys today. So if you have any questions, shoot me some in the chat here.
Awesome. Thanks, Paige. That's great. Yeah. I always say that Paige, like she's got to work within the bug schedule and then move east to west, working on the plant's phenology. And it's a real test in adaptive management. I think it's delivering on that biocontrol release program. It's logistically complicated, but Paige does a great job. So yeah. Any questions for Paige? Yeah, there we go. So Paige asks, are any of the agents suitable for release in floodplains? Do you want to talk about floodplains a little bit, Paige? And why? Yeah.
Yeah, we don't really do, if we try to release above the floodplain or high water marks so that any of the agents like leafy spurge beetle and the root weevils for spotted and diffused knapweed will overwinter underground. So we don't want to kind of drown them out. I think you could probably get away with it for maybe a Russian knapweed release or the seedhead weevils, but I wouldn't strive for that. I think we just don't want to risk them kind of dying.
It's tricky for sure. Yeah, you want to set them up for success as much as possible, right? And there's a lot of factors involved in even just getting them to establish. And some of it's hit and miss too. Hey, Paige, like you see a perfect site, you do the release and they don't do well. And it's tough to know exactly why, but
totally. Yeah.
Hi, Nathaniel. Nathaniel asks, and this might be more for Phil. So Phil, you did mention Pilosella and some hawkweed. So we've got some hawkweed problems here in Alberta. Nathaniel asks if the Pilosella species was rolled out already for hawkweed. Is that just in the US for the agents? Okay, Phil has typed a response. Do you want to?
I had to answer because I just finished. Yeah, so there is Kilosia urbana, which is the root mining fly. We actually just sent another shipment of eggs and adults a couple of weeks ago to Chandra Moffat in Summerland in BC. And she's working on getting the fly established in Canada. It's been a challenging species to work with. She's made We really struggled. Firstly, we struggled to get material alive from Europe to Canada to get
Mm.
kind of established on plants. We spent a couple of years figuring, we're also a very short window. They're only active, and Paige will know what I'm talking about when I say this, but only active for like a couple of weeks of the year as adults. So we can only collect them then, and then it's, phenology in Canada is not the same as Europe and so on and so forth. We worked quite hard and we figured that out. In the last couple of years, we've sent several hundred eggs over, and we're hoping to get establishment in the next couple of years. And then I'm not sure though. You'd have to check with Chandra herself and maybe Rose to Clerk Float where the releases are actually happening. I know that there's some happening in BC, but it's quite possible through the Lethbridge Center or Institute or lab, whatever you guys call it over there. There may be some releases in Alberta as well.
And what hawkweed species are the agents for? Do you remember? I think it's like meadow or
Mouse
mouse
ear.
ear.
You're catching me on the spot here. To be honest, I'm not super confident.
That's okay.
There's a couple of species, and I I'd have to get back to you on that. I don't
No
want to say
worries.
something that's not right, but I think it's mouse ear is the Kilosia urbana, and then the wasp that's being petitioned for release, I think, is for orange. But
Oh, that'd be
I
good.
can get back to you, Megan. You can send that out to the group.
Okay. Yeah. Perfect. Thanks, Phil.
Yeah.
Great. Awesome. Do we have any other questions for Paige or Phil? Okay. I don't see any. And again, there will be opportunity at the very end. So I'm going to invite Erin back up now to talk about the concern. And Erin, do you also want to mention the comment that you had about leafy spurge? Because that was a great comment. Yeah. Did you want to just talk about that a little bit?
Yep. Yep. Well, thanks for inviting me to this. Rick emailed me about this. We met in Annapolis the other day and had some good conversations. So I appreciate the opportunity to share with you guys how Wyoming does biocontrol and gets agents on the ground. So my name is Aaron Foster. My day job is I'm the district supervisor for Fremont County Weed and Pest District it's in Lander, Wyoming. So kind of central northwest-ish part of the state. And I'm also the chairman for the Biocontrol Steering Committee for the Wyoming Weed and Pest Council. And I'll get to a little more detail on that in a minute. Going back to the comment, I was going to chime in and then my timing was off, so I just threw it in the chat. But there is a study that was conducted by my predecessor and a past employee here at Fremont County for impacts to a native spurge called Euphorbia robusta. And they observed that there was some feeding going on between the two species, the leafy spurge, invasive, and the native robusta, because they coexisted in the release area. And so they were concerned with that, and they looked into it. And to correct what I mentioned there, concluding that the feeding was incidental, what they really concluded on was that the biocontrol agents reduced the population of the leafy spurge so well that the robusta populations increased as a result. So that's really how it is. And I shared a link with you all for that kind of a summary of that presentation that was given quite a few years back somewhere. I don't even remember where. But if you're looking for more information on that, I could probably dig in and find a little bit more onto it if you're interested in it. So that's that. Any questions on that before I move on to Wyoming Biocontrol Steering Committee? Okay. So I didn't prepare a slideshow or anything. I was just going to talk to you guys a little bit about Wyoming's Biocontrol Consortia and back and forth discussion if you had any on how we do things and how maybe you guys could adapt some of that if you wanted to. So I guess just a little bit of history on us and some things. So Wyoming is fortunate. We're kind of unique in a lot of ways in the states as far as how our weed and pest districts are funded and formed. A lot of states have weed management programs going on, but a lot of them don't have the funding mechanism stability like Wyoming does, where we have a specific tax for the weed and pest district for every county or district within Wyoming, specifically for these purposes. So that's a stable funding source that helps keep us going and providing services to our local community for invasive species management. And that got established in 1973. Since then, we got interested in biocontrol as well. Our first biocontrol agents were released for musk thistle control in 1978. And we were noticing that that was a positive result associated with that. So we organized some different efforts to try to get biocontrols for musk thistle, additional agents, and leafy spurge was the focus at the time. The first release of the flea beetles for leafy spurge control, it was in 1990, and it was soon noticed that those insects were having a pretty significant impact on the populations of leafy spurge around Wyoming. So the Biocontrol Committee was formed at that point with the primary purpose of identifying leafy spurge that occurred in every section of Wyoming and release the agent in those sections. And that's what the Biocontrol Committee did at that time. It was a successful committee, and it has kept going since then and has adapted to taking on other goals and purposes, such as supporting biocontrol, overseas research and development. We also get some funding from a grant from Wyoming that we've had from the Wyoming Department of Ag we've had since 1994, in addition to other sources. So our Biocontrol Committee is a component committee of the Wyoming Weed and Pest Council, which Wyoming Weed and Pest Council is comprised of members from every district in the states. All 23 counties are members and participate in that. And we also have representation from the Department of Agriculture, University of Wyoming, and then our partners with some federal agencies and such. The committee is comprised of 6 voting members. Each member represents an area of those 23 districts. But we also have those partners with the University of Wyoming and APIS involved to help direct our activities. But we look for participation from all the other counties and any other interested parties. The mission is to help facilitate the research and implementation of biocontrol projects in Wyoming. And our goals, 5 main goals I'll talk about here. First one is to help development and distribution of agents that are at insectaries within the state. So, for example, a fairly new one is the white top mite, and we're trying to get that established in Wyoming and then be able to use that insectary to distribute to other counties. And then we try to facilitate the introduction of new agents into the state. So the white top is another example of that. It was in Montana, and we wanted to get some here so we could start on insectary. So that was the committee's efforts to try to get some of that into Wyoming first and move that around. And then our third priority is to contribute to these consortiums that do that sponsor biocontrol research and development, with CABI in particular. We also try to aid districts. Some of our districts, like I said, have stable funding, but the funding is dependent a lot on their resources. So if the county has minerals, they're rich. If they don't have minerals, they can still be pretty poor. So we try to help get some of those agents, to help them purchase agents and get agents when they don't have the funds to do it themselves. And then more recently, we're also trying to advocate and promote monitoring of biocontrol releases utilizing the standard impact monitoring protocol. One was kind of late to the game in that. We're trying to ramp that up. So we've had success with biocontrol in Wyoming. We've released over 30 agents on 10 different weed species. We think that about 20 of those are well established and have an impacts at the population level. Others are established and maybe not as impactful, like the Canada thistle, weevils. And we feel like biocontrol is a successful tool overall. Let's see what else I want to talk to you guys about. So the majority of our funding definitely goes to CABI. Historically, Wyoming has had a lot of support from the different counties contributing to the consortium. So we get, like I said earlier, we get funding from the Department of Agriculture through a grant, and then the bulk of the funding had been individual districts, counties contributing to the Biocontrol Consortium for overseas research and development. When I got started as the chairman, which was I think 2012 or so, we were consistently funding somewhere in the range of $250,000 to $300,000 to CABI a year. And recently that has declined. Now we're contributing about $112,000 a year. The biggest driver of that reduction in funding was driven by a stall in key personnel with the Fish and Wildlife Service, who reviews the petitioning. As Phil talked about, our process is a lot more arduous than it seems Canada's, which I'm envious of you guys for that. So there was a key position in Fish and Wildlife that was vacant, and so petitions were just being stalled and the queue was building and there was no progress being made. So we were putting all this money into research, but there was no fruits from that labor. And so a lot of the weed and pest districts in Wyoming said, you know what, I feel like this is going nowhere. I'm going to pull my funds. And that symptom is still in play today. We still can't get some districts who have historically contributed a lot to contribute. However, we still do have a fairly decent amount of funding for CABI. We're probably one of the leading contributors still, I would think. From my understanding, there's not a lot of states that are contributing to biocontrol research in the U.S., at least to CABI. I think there's 4 of us. Montana and Wyoming are kind of two bigger ones in the West. With that, you know, Wyoming's projects we're focusing on right now are the Dalmatian and yellow toe flax biocontrol projects. Russian knapweed has been a focus for ours for a while. White top, oxeye daisy. So I'm excited that you guys got the oxeye daisy insects up in Canada. We're waiting on that. Dyer's woad is another project Wyoming has a lot of interest in. Houndstongue and Russian olive. Again, another one that we're envious of you guys. That's currently stalled in our process here in the States through a unique request that has never been initiated before, from my understanding, with tribal consultation. So the tribes are requesting this, and that's kind of stalled that process at the moment. We're definitely interested in other projects too. We just don't have the funds to support it. So, you know, groups like yourselves who join into this effort and help, you know, stretch the dollars further for different projects are going to benefit us. You know, we're interested in common tansy. There has been a little bit of interest in blueweed. We don't have a lot of that in the state, but there are some places that are having problems that would love to see some biocontrol. And then for us, in Wyoming anyway, annual invasive grasses such as ventonata, medusa head, cheatgrass are significant problems that we want to see some biocontrol on if we could. So that's kind of my overview of how it all works. So just to kind of summarize again how ours works, we have a committee, we have bylaws. I sent those bylaws to Rick. If you guys ever want to look at that, I can share with anybody that wants to see that. But our committee is formed. We have some representation that makes decisions. We get funding through sponsorship from different counties and districts and interest parties, and then we get a grant as well to help pay for this effort. We have had outside sources contribute as well. We've received funds from a county in Montana for the Russian olive biocontrol over the years, because their state didn't have a mechanism for that. So they reached out to Wyoming and said, hey, can we throw you guys some funds to support the Russian olive work? And we said, absolutely. We just send them a bill and they give us the money, and we put that to use towards CABI. So we have a history of that, and I believe we've done it with California too for one incident in the history of it. So I think that's really all that I had from our perspective, but happy to answer any questions if you guys have it about our Biocontrol Steering Committee.
There we go. Thanks so much, Aaron. That was great. So how many organizations contribute versus the steering committee, or like the actual committee? What does that kind of structure look like, and the numbers look like there?
You know, Phil might be a good person to ask that question too, but I can tell you one example. The White Top Biocontrol Consortium, from my understanding, correct me if I'm wrong, Phil, has been sponsored by Wyoming Biocontrol Committee, Montana Biocontrol, the British Columbia BLM, and I think in the United States, USDA has also contributed to that.
Nice.
Yeah, USDA.
So there's several.
Awesome. And then within your own, like just your Wyoming program, like how many counties are like involved with?
Oh, okay. Yeah, currently we're probably looking at just like 5 or 6 of us that are actually actively contributing.
Okay.
Several are participating in the committee, but don't actually sponsor funding at the moment. You go back in the time with it, we had more, like I mentioned, we had more support. So since 2009, all but 3 counties have actually contributed financially to the Biocontrol Consortium at some point in time or another, and at some level or another. So, the poor counties and the rich counties, there's discrepancy there. So we've had contributions of $500 a year that come to the consortia.
Yeah.
Like some counties are like, if I have $500, can I give that to you? Absolutely. To others who really feel strongly about this and have lots of funds coming from mineral development. So they, have one that's done to $70,000 a year.
Wow.
So yeah,
Okay.
it varies widely. Fremont County, for example, my county, I contribute $10,000 a year, and that's gone down. I used to do about 20. So that's where we are.
Yeah.
That makes sense.
That's awesome. Yeah. No, fantastic. And are there other lots of like very detailed questions, but are there other
Yeah.
like external other than counties, like other people contributing within Wyoming? Like, do you have industry groups or other NGOs or anything like that? Just a county level kind of financial contribution. Hey?
It's primarily been county level. We've tried a little bit to tap into like conservation districts. I don't know if you have something
Yeah.
similar in Canada. And that hasn't really panned out too much. We weren't too aggressive with it yet. And we haven't tapped into industry as much either. There's oftentimes a perception from land managers that, biocontrol doesn't work. But I think the perception comes from their experience with herbicides, right? So they see an herbicide kills the weed.
Yeah.
Biocontrol, there's still weeds in the landscape. And so it's hard sometimes to convince them that it's a valuable tool, even though success is there, and we just need to balance our expectations
Yeah.
with biocontrol and the success that we get from it. And that's something Wyoming also is trying to educate folks on that a little bit more. So to answer your question shortly, no. We have had outside sources, like I mentioned in my talk a minute ago, coming from other states like Montana. We had a Liberty County, Montana, contributed to our Russian olive consortium. So we've opened the door for that. We've actually offered that to other folks too, if they don't have a system. Hey, Wyoming could funnel it through if you want to send it their way. No takers.
Interesting.
Yeah.
Yeah, and that's an interesting thing too. And I mean, a good point about managing expectations. Like none of this is going to happen overnight, right? This is slow. The process in developing is slow. Biocontrol, when they're actually you have them developed and you're releasing them, it can be slow too, right? Like it's not like you spray it and it dies the next day. So I think that's an important part of this too. I do think that there is some, ESG money, like these companies that have money that they want to put towards, environmental initiatives and nature-based solutions. And I think that might be an opportunity maybe for us here in Alberta, particularly for us here to tap into. So, yeah, that's fantastic. Any other questions for Aaron or Phil or Paige? Okay. I'm not seeing it. Oh, Paige is back. Perfect. Yeah. So I'm not seeing any. So I guess what I'll do now is just kind of provide just a general update on the progress to date. So, so this was really Rick's idea, Rick Niwa from MD of Ranchland. He loves being the center of attention. So I'll just bring up his name all the time and maybe make him turn on his camera. I don't think he wants to do that either, but that's okay. So this is really Rick's brainchild. So he was so this is how this came to be, was that he was talking with some industry partners and colleagues down in the South here about potentially contributing to something along the lines of a biocontrol consortium and funding biocontrol research. Because again, it's kind of within that that ESG framework that they have and what they want to do to look sustainable. So we met, Phil and Rick and Paige and I all met up at NASMA in the fall, and we had a discussion about what this could potentially look like, and we gained some more information to learn more about CABI and what the process looked like. Had a great meeting then. And then once we came back from that, Rick and I worked on sending out some communications to the AAAF regions. So all of the counties within Alberta, and we got a representative from each of the regions, and we asked them to help us shortlist priority species for biocontrol agent development, is basically what happened. And so there will be a lot of discussion, I think, with our priorities with what Cabby already knows, the knowledge base that Phil and his team at Cabby already have, and understanding, you know, the work that's already been done, what are good potential weeds and that sort of thing. But basically wanted to kind of get some information from our counties to figure out, you know, what the priorities were there as well. And then in March at the AISC conference, Rick introduced the concept of an Alberta Biocontrol Consortium at the conference. So I know many of you were there. And so Rick had an opportunity to kind of provide this bit of outline at that event as well. So there's been a little bit of kind of work and momentum happening all along. And then Rick and Shayna just attended the National Biocontrol Forum. I think Phil mentioned that down in the States. I think that's another NASMA event. And that's where I think Rick connected with Aaron and learned of the Wyoming Consortium. So lots of learning from our neighbors and colleagues and people that have already been doing these things. And then today, of course, we're having this webinar to really kind of hopefully bring everyone's understanding of biocontrol, how the agents are developed, and just the general process and timelines and costs, et cetera. We bring our awareness all up so that we're all on the same page, and then figure out what our steps are going to be moving forward. So I do have a QR code with a form. Hopefully it works. I try to do these things, folks, and it never works. But let me just test this this form out. I did test it before, but sometimes it just stops working. So if you're interested, if you've learned today and you're interested in kind of keeping the communications open, then please enter your information on this form. It works. Yay. It's a miracle. It's wonderful. So please enter your information in that form. And that does not mean that you're committing to anything. It just means that we'll keep you in the loop and we'll let you know what the next steps are. And in our mind, what the next step will be is to set up a meeting with everyone that's interested in being involved, and to work within that group to set up a terms of reference so that we all kind of understand what kind of framework and kind of, you know, criteria and what this kind of partnership is going to look like, and then kind of figure out all the details from there. But I do encourage everyone to sign up, to be kept in the loop. Again, this is not committing yourself to being a part of anything. It's not committing yourself to contributing funding, but just to being kept in the loop. And then we'll reach out to that group, everyone that signs up through this form. We'll reach out to you and keep you in the loop, let you know when the meeting will be held, let you know and invite you to join that meeting if you should choose to do so. But again, no commitment. And I definitely encourage everyone to sign up for that. Or if you're having issues with the form and a QR code, just send us an email. I think most of you know how to track down Paige and I. So feel free to do that as well.
I will say the form says default with a star. I think that is the spot for email, because right below it says reconfirm. So email on those bottom two spots.
I think there was a message in the chat about that as well.
Thank you, everyone. See, I shouldn't be allowed to do these things. Thank goodness for Paige. Thank you. There we go. Okay, good. Well, we'll get it sorted one way or the other.
Well, at least the QR code worked.
Yeah, that, you know. So I will leave the QR code up for a little bit longer, but folks, does anyone have any questions? Is this or comments? Is this something that you're interested in being involved in? What are the needs and priorities in your area? How might this fit within your programming? Anyone have anything that they want to contribute before we wrap up here? Okay, just give it another play the Jeopardy music. But okay, so I'm not seeing a lot of other questions. So what we'll do, again, make sure that you sign up in the defaults. Enter your email into the default section, or fire an email to Paige and I. Will get you looped in. And again, we're just going to start communicating about this and figuring out what those next steps will be. We'll convene a meeting. Awesome. Thanks, Allie. Allie says she's a seasonal worker with a municipality. The topic's very interesting. Absolutely. We really need to work on developing more tools and resources for battling weeds, right? And I think it's about being kind of proactive and getting, just working towards developing these other control methods. And Sarah said, sounds great. Parts of our team will reach out. That's awesome. Thanks so much, Sarah. Okay. Rick, did you want to, not to put you on the spot, Rick, but do you want to add anything before we sign off?
I just wanted to thank everybody that presented today. Thanks, Aaron, so much for yours, and Phil and Megan and Paige. It was great.
Oh, thanks, Rick. I'll echo
Thank
that.
you.
Thank you. Yeah, thank you,
Absolute
particularly
pleasure.
Rick. And our international meeting here this morning, it is again, it's now after 6, I believe, on Friday evening for Phil. So he's probably itching to get off and dive into the cheese fondue. I think that was
Yeah.
your next activity, right? So yeah.
It is indeed. I mean, I think maybe just as a closing statement,
Yeah.
I think with biocontrol, it's quite important that we stay on the wagon, if that makes sense. So, a lot of the successes that we're experiencing now are from work that was done, anywhere from 10 to 20 years ago. But if we were to stop, we would end up having this lag. And I think that's what Aaron kind of alluded to a little bit with the regulatory situation in the US getting so tough, is that there's actually been now a 10-year gap, if not longer, actually, in biocontrol agents getting, so they've been ready and petitioned and essentially ready to go, but they're just stuck in the kind of permitting process. And, don't kind of, I think what I'm trying to say is that we need to continuously so weeds that we target today may only become a success in 20 years when, we're all retired. But think about your children and your grandchildren, and so keeping those agents going and invasive species under control is an ongoing battle that we have to keep going. And yeah.
It's kind of like planting a tree right now.
Yeah,
You might
exactly.
not reap the benefits, but yeah, exactly. Yeah.
Absolutely.
Yeah. No, for sure. All right. Okay. Well, thanks again, Phil and Aaron, so much for joining us. We'll be back in touch with you guys, I'm sure, to get more information. Thank you, Paige. Excellent overview. Yeah, we sure appreciate everyone's time in pulling this together. And again, we'll be back in touch with everybody as well. So thank you, everyone, for joining us. Have a great Friday, and we'll be in touch. Great. Thank you.
Thanks, everyone, for pulling this together and for all your time as well.
Thanks, Phil.
Cheers.
All right. Have a great day, everyone. Thanks. Bye-bye.